Alright, welcome everybody. We're doing Genetics Part 2 today. Genetics Part 2 is going to take a look at the concepts of gene expression and mutation. I'm going to do this in two parts for Part 2. So we'll have Genetics Part 2.1 and Part 2.2. In 2.1, we'll do gene expression. In 2.2, we'll talk about mutation. So let's go ahead and get started. Let's talk about gene expression in Part 1. So, first question is, what is it? And in general, it's the process by which a gene, the sequence of DNA base pairs, is made into a gene product. And we've talked about this in Part 1, but the gene product is going to be some kind of protein or RNA. For the most part, in our lecture today, we're going to focus on the roles of proteins. So, in other words, we talked about the terms genotype and phenotype in Part 1. This is essentially when the genotype becomes the phenotype, or you say it as the genotype is expressed into the phenotype. During gene expression, DNA is first transcribed into RNA and will then later be translated into proteins. So here in just a second, we're going to take a look at how these terms transcribe and translate relate to the concept of gene expression. But just to reiterate this point one more time here, we're essentially making a gene product. This is going to be protein or RNA. Your genotype contains the genetic information, and that genetic information is expressed or becomes whatever phenotype that gene codes for. Now, we've talked about the central dogma a lot already this semester, and we talked about it in Part 1, but I want to now sort of connect that to these the concept here of gene expression. So we're going to use the central dogma to diagram the process of gene expression. So here we've got the central dogma drawn up, and we'll make this pretty quick, but what you want to do is recognize that the first part of the central dogma, DNA to RNA, this process is the first stage of gene expression, and this process is referred to as transcription. So transcription is essentially the conversion of DNA into RNA. So this is the first stage of gene expression. The next stage, RNA into protein, this stage is referred to as translation. So the gene expression essentially follows the central dogma of biology, and it does so in two distinct stages: transcription, the conversion of DNA to RNA, and translation, the conversion of RNA into protein. And so we're going to look at this in two parts. We're going to start with transcription, and then we're going to look at translation. So this is how the central dogma can be used to diagram the process of gene expression. Before I go any further, I would like to strongly encourage you to watch a video that you can find on YouTube about that shows the animation of DNA transcription and translation. Now, there are more than one, and you don't necessarily have to watch this one, but I think this is the best one I've found so far. And if you find anything that you think is as good or better, then let me know, because these things always change. So this is as of October 8th, 2019. So things are always going to change after that. But this video has been up for a couple years now, and I think it does the best job of really showing all the steps without going into too much detail. It's only about 7 minutes, and it shows really good, clear animations. I highly recommend that you watch this video before going any further, and it will go all the way through translation. So at the very least, watch the first part of it for transcription, and then go back and watch the second part for translation when you get to that part. This will make it much more clear what I'm talking about when you watch the video first. Okay, and I would recommend watching it a couple times, but I will put that up one more time. So just go to YouTube, type in DNA transcription and translation. And this, by the way, is from McGraw-Hill Animations. So I can't really show the video in my video, but I can definitely tell you to go watch it here. So McGraw-Hill Animations, DNA transcription and translation. Okay, moving on here. So what is transcription? Transcription is a process by which DNA is converted into RNA. Now we're going to get more specific. So the purpose of transcription is to convert the structural genes into RNA intermediates. Recall that a structural gene is a gene that codes for protein. So the idea is your DNA holds the information for how to make protein. This is something we've talked about. And so during transcription, we're essentially starting the process of expressing that gene into the physical protein structure itself. So we're taking that and converting it into an RNA intermediate. So RNA will physically carry the information for protein synthesis out of the nucleus and into the ribosomes that are located in the cytoplasm. So during this process, ribosomes will utilize the RNA to then synthesize proteins. That will come up in the concept of translation. So transcription is taking the DNA, converting it into an RNA intermediate. That's the starting idea. There are a number of things involved in transcription. It's a fairly complex process, but we're going to focus on the major participants. So the major participants, and there's a couple, but the big one is an enzyme called RNA polymerase. This is an enzyme that's responsible for the synthesis of the RNA itself. Now this actually works by using the DNA as a template and synthesizing a new strand of RNA using the DNA itself as a template. And this is going to look a lot like how DNA is replicated. In part one, we talked about DNA polymerase and how DNA polymerase replicates the DNA by using one side or the other as a template. This process is very similar. The names of the enzymes involved are very similar. RNA polymerase versus DNA polymerase. So they have similar names and similar roles. So they utilize the template to make a new strand. Complementary base pairing occurs between DNA and the RNA. So recall from part one, I talked about the complementarity between DNA itself, and then I talked about the complementarity between DNA and RNA. And so in this case, the complementarity between DNA and RNA will apply. So it's going to look something like this. The DNA itself will be opened up or unzipped. And the one side or the other of the DNA molecule will be used as what's referred to as a template. So we only use one half of the DNA molecule, one side or the other. So whatever side's being used, that side of the molecule will be acted upon by RNA polymerase by taking the information on that side of the molecule and using complementary base pairing rules to actually build a new RNA version of that information on top of the DNA template. And so then this process will finish at some point downstream from where it starts, and this RNA molecule, as we'll see here, will dissociate and become a free-floating, single-stranded piece of RNA. So we'll go through those steps here next. So transcription takes place in 3 stages: initiation, elongation, and termination. Initiation is an interesting process because initiation involves a series of molecules that act to regulate when and where and how frequently the transcription process takes place. So this actually is done by what's called a promoter region and what are called transcription factors that we'll get to next that control essentially the timing and the frequency of protein synthesis. So these act as a way to regulate the cell to only produce proteins upon demand. But that's the idea of initiation, and ultimately it's just the starting process, but it's also a regulatory stage. Elongation is the point at which the RNA polymerase actually starts to make a new RNA molecule using the complementary base pairing rules. And termination occurs when we reach a sequence in the DNA that is a recognized stop point. And we're going to simplify it down for this class. There's actually different types of termination sequences and different ways in which that can work. But for simplicity, we're going to just say that that's the point that it ends. Okay, so we're going to focus more on initiation and elongation here. So initiation, as I mentioned, is the start of the process, but it's also a regulatory point in the process. Not every protein is going to be produced automatically, and not every protein is going to be produced with the same frequency and intensity. Some proteins are produced all the time, some proteins are produced very rarely. And so oftentimes when and where and how much of that synthesis is taking place can be directly or indirectly controlled by transcription factors. So they actually have to bind to the DNA molecule somewhere upstream from the gene. And that binding of transcription factors will then facilitate the actual transcription process to initiate. So they bind to DNA and facilitate the binding of RNA polymerase, which will then allow elongation to take place. So without transcription factors, initiation of transcription cannot occur. So they not only initiate it, as I mentioned, but they actually are a way to regulate gene expression. And so we're simplifying all the details down, but you essentially have a region in the DNA where the information is being transcribed. Upstream from that, we have different regions that act as control points. So different molecules, transcription factors themselves, have to actually bind to these regions in order for transcription to take place. And in some cases, they'll completely prevent transcription from occurring if they're absent. In other cases, they will slow transcription down so that it's not happening as frequently. And so not only can they turn it on or off like a switch, but they can also turn it up or down more like a throttle. And with that throttle, then protein transcription can be increased or decreased. So this is a really significant way in which the cell controls how much when and where proteins are being produced. Now, we're not going to get into what exactly type transcription factors are, so we're going to keep that concept broad. So just recognize they are molecules that bind and regulate transcription by controlling the initiation. So the truth is, this is a pretty complex thing because every gene can have different transcription factors, and the specifics involved can vary from one gene to the next. And so we're going to have to keep it broad for a class like this. So that's the idea behind initiation. Initiation will occur at a site called a promoter region. A promoter region is the specific area where the transcription factors will bind. So as I mentioned, they occur upstream. So if this is the transcribed region, this is the area that we're going to elongate into an RNA molecule. The promoter region will occur just upstream from that. From there then, RNA polymerase will bind to that and move downstream to turn that into the RNA product itself. The promoter regions are often known to have regions that have specific sequences of thymines and adenines. And because of this, they're often referred to as a Tata box. So the Tata box is a specific region, a specific promoter region where initiation often begins. Often, but not always. So here you can see that specifically in this diagram, the Tata box, the transcription site downstream from that. So once initiation is complete, RNA polymerase will start to move along the DNA and synthesize an RNA molecule using the complementary base pairing rules between DNA and RNA. So we're elongating the RNA molecule by synthesizing one base pair at a time, or one nucleotide at a time. Only one side of the DNA molecule is quote-unquote read during elongation. So this is one of the confusing things about DNA structure. You have two sides of a molecule, but only one side or the other is actually transcribed. Now, it can vary as to what side is transcribed, and this is actually a much more complex thing. But in simple, only one side is transcribed, and whatever side that is, this is known as the template strand. The other side is going to be considered the non-template strand. So it can switch sides, but in reference to whatever gene and whatever side that gene is being transcribed by, you have the terms template and non-template strand to correlate. So that would look something like this. Here we see the DNA in the RNA polymerase. It's moving along and synthesizing an RNA molecule, and it's doing so on this side in the figure, making this side the template strand and this side the non-template strand. And as you see here, the RNA is being created using the DNA as a template, following those DNA to RNA base pairing rules. So this allows this specific genetic information to be transcribed into a specific RNA molecule. So transcribing is sort of like taking one language and turning it into a different language. So if I was to write Arabic into English or English into Arabic, if I'm writing that down, I'm transcribing from one language to the other. And so this is sort of like how DNA and RNA are working. They are essentially saying the same thing, but you can think of them as two different genetic languages, the language of DNA and the language of RNA. And so during transcription, we're literally converting that in the form of DNA into the form of RNA, like converting from one language to another. So elongation will take place in reference to how large the gene for that protein is. And so different genes have different sizes, but ultimately elongation occurs along the length of that gene. At some point, we'll reach what's called a termination sequence. And a termination sequence is basically just the point at which the gene stops. The newly synthesized RNA will then dissociate from the molecule, and following this, there will be typically some kind of processing that occurs. In other words, the RNA has to be sort of polished in order for it to actually move on to the next stage. This is something that varies. In eukaryotes, a sequence of adenine nucleotides is often added to one side, and what this does is it preps it to leave the nucleus. Usually it can't actually get out of the nucleus where transcription occurs until it has this poly A tail on it, and then from there it can be acted upon in a way to move out of the nucleus. So the poly A tail signifies that the RNA molecule is ready and ready to leave the nucleus and enter the cytoplasm. So here's a figure showing kind of what that looks like. Once the termination sequence is reached, the RNA itself is usually starting to bunch up as it becomes a long molecule. As I mentioned, there are actually a couple different ways the termination can occur. Sometimes it actually dissociates itself due to the size, and other times there are specific molecules that help pull it off of the DNA. So we're sort of skipping some of those specific details here, keeping this as short as we can. But ultimately, it reaches a point where the gene is no longer being transcribed, and the RNA itself disassociates. And then following disassociation, things like a poly A tail are added to one side, and that occurs with special enzymes external to RNA polymerase and external to the DNA template. Okay, so we've said that the transcription process is the conversion of DNA into RNA. And now we're going to get just a little bit more specific. So the specific product of transcription is a type of RNA referred to as messenger RNA. Messenger RNA is essentially named because of the fact that it carries the genetic message on to the next stage of gene expression, which is translation. So the genes themselves carry information for how to make a protein, and the message for how to make that gene is in the form of a specific kind of RNA called messenger RNA. So recall from part 1 that there are different types of RNA. And so in the specific process of transcription, we're making a type of RNA called messenger RNA. Now, messenger RNA is significant in that its sequence of RNA can actually be broken up into segments referred to as codons. So in other words, its message is actually specifically worded, quote unquote, in a form called codons. So codons carry the code of DNA, so to speak. And codons come in a format that are based upon 3 nucleotides. And for the most part, each of these codons are going to code for specific amino acids. So think back, what are the building blocks of protein? The building blocks of protein are amino acids. How do you make a protein? You make a protein by joining amino acids together to form the chain of amino acids that then folds. This is what we talked about in chapter 7 and 8, metabolism and growth. Ultimately, the message in the RNA, the messenger RNA, spells out this sequence of amino acids, and those amino acids are specifically spelled out by what are called codons. And so I'm going to get to that next here. There are some codons that are actually designated stop codons, and they don't code for amino acids. So I'm going to break that down here shortly. But codons, for the most part, are code for the amino acid sequence. And so we see here DNA being converted to a specific type called messenger RNA. And if we look, as we'll see here next, each 3 nucleotides actually signify an individual codon. So we can see that here a little more clearly on this figure. Messenger RNA, each codon translates into one of the 20 amino acids or is a stop or start signal. So we'll get to that part on the starting and stopping here. But take a look. Here's a sequence of RNA. In these RNA molecules, RNA nucleotides then are shown in their individual codon units. So codon 1, 2, 3, 4, etc. And then down below here, what that's saying is that the codons themselves are actually signifying or specifying particular amino acids. So like codon 1, for example, is specifying the amino acid methionine. Codon 2, glycine. Codon 3, serine, isoleucine, etc. Until we get to the end and we have what's called a stop codon, and that signifies the end of the protein chain. So when it is said that DNA codes for protein, it's actually a little bit more specific to say that DNA codes for the amino acid sequence of a protein. That's actually really what DNA is coding for. How do you build the protein one amino acid at a time? So think of it, if I said I have a cookbook that tells you how to make a meal, specifically what that cookbook is going to tell you is the step-by-step sequence to make that meal, right? And just sort of nuance into exactly what we're saying. But when you get down to the specifics, you know, the cookbook or the recipe itself is telling you step-by-step ingredients and the things that go into that recipe, into that meal. So this is what we're looking at with messenger RNA. So it carries the information for the sequence of codons. Just a little bit more specifically, the sequence of codons specifies the type, the number, and the order of amino acids. So we have 20 different amino acids. Each one is a different type. So at any point in this chain, we could theoretically have any of those 20 amino acids. So that's the type. Different proteins will have different numbers of amino acids. So some proteins may have only 100 amino acids, and some proteins may have 1,000 or more amino acids. So how many is also specified in the size of the messenger RNA and the size of the gene itself. From there then, what order do they go in? Okay, so from each of these then we end up with a specific protein chain that will ultimately fold into its three-dimensional shape depending upon how these amino acids are arranged. So we didn't get into all the biochemistry of protein folding, but you just need to understand that this exact sequence will have will have all the significance that it needs to allow the protein itself to fold into a three-dimensional shape. So if we switch these orders around, if we take amino acids out, the protein will fold differently, and that will lead to a different shape, and that will lead to a different function. So ultimately what we're doing here is we're actually spelling out how the protein will ultimately fold indirectly by coding for the type, number, and order of amino acids. Now this is a figure out of the text that shows all the different possible codons and what they actually translate to in terms of the amino acids, and some of these being official stop codons. So there are 64 total codons. If you count them up, we've got 64 here. And if you do the math on it, there's actually only 64 possible units that you could have. If you have only 4 base pairs, and this is showing RNA here, so U, C, A, and G. If you have 4 base pairs, and we have 3 positions we can take each base pair to, that is 4 to the 3rd power. And so that gives you the number of different combinations you can have with 4 units and 4 nucleotides with 3 positions for each nucleotide. So if you take 4 to the 3rd power, which is 4 times 4 times 4, that gives you 64. So we actually have the exact number of codons that you can possibly have. And if you look here with our 4 RNA base pairs, U, C, A, and G, this gives you all 64 of those possible outcomes. So of those 64, we have one that is officially a start codon. This codon here, in the vast majority of cases, is always going to occur at the very beginning of the RNA molecule. 3 are official stop codons. So those are shown here, UAA, UAG, and UGA. These are all official stop codons. They would always occur at the end of the messenger RNA. All of the rest of them code for one of the 20 amino acids. And as you can see here, many of them code for the same amino acid. And we'll come back to that concept later. This is called degeneracy. So we have more than one codon sequence coding for the same amino acid in many cases. So the only exception is the start codon for methionine here. But the rest of these have at least 2 or more different codons that code for the same amino acid. So for example, UUU and UUC both code for phenylalanine. And for the amino acid serine, we have 4 different codons that all code for that same amino acid. So this is called redundancy. And we'll talk a little bit later about why that's important. But that's the idea behind codons. So going back one more time here just to visualize that again, this is your sequence in the messenger RNA. And that sequence then is going to connect to that chart there, telling you which amino acid goes into the final protein chain based upon this codon, based upon how that codon maps to the amino acid. And it's probably worth pointing out that this is something that took scientists a long time to figure out. And what it basically shows us is that there is actually almost literally a universal language when it comes to the way cells actually express themselves. And so this is one of the most difficult things to wrap your head around when it comes to just a biology class, but also when you actually start to realize that this is a core process to all living things. All living things, no matter whether it's a bacteria or whether it's a human or anything in between, actually speaks the same language. These codons here map out into proteins in all life forms, no matter what kind of life form you are. It's an amazing thing. And so what we're really actually looking at here is not just some technical terms, but an actual language that is expressed by all living things. And so in the DNA is information ultimately for how to build a protein. And this is actually sort of like the manual. If you were to put together a coffee table and you had to pull out the manual and read the directions step by step by step, this is sort of like the equivalent of that manual. Here's the instructions. here's, if we see this sequence here, that means we put this amino acid here. And then the next amino acid is this, etc., based upon all of that coding information. So it's kind of worth stopping for a second, pointing out that it's not just a technical list, but actually sort of the instruction manual for all life. It's a pretty incredible thing, actually. Okay, so next here then, in eukaryotic organisms in particular, once the RNA molecule is produced, you have a series of steps that occur after that, referred to as RNA splicing. So this is specific to eukaryotes. And what happens is the RNA will go through an extra series of steps in which the what's called the premature RNA is modified into what's referred to as the mature RNA. This part gets a little confusing, but premature RNA essentially will contain extra regions that are not needed in the final protein sequence. These regions are referred to as introns. Introns are non-protein coding regions. Other regions in the RNA are called exons, and these are the protein coding regions. Let me skip forward here real quick to the figure I had next. And what this shows us, we have our DNA template, we have our transcript, the messenger RNA. This is the premature form here. And within this premature RNA, we have regions of coding and non-coding sections. So Mother Nature has it so that when the RNA in a eukaryote is produced, it actually needs to be processed before it goes on to the next stage, which is translation. And so what happens is these little enzymes called spliceosomes will actually cut out certain regions, shown here in green, and glue back together, so to speak, these regions here shown in red. So the introns are actually cut out and the exons are glued back together. And this process is called RNA splicing. So during splicing, introns are removed and exons are joined together to make the final mature RNA. The enzyme involved, well, actually, it's not an enzyme. It kind of is. It's actually just referred to as a molecular complex. But I won't trip you up on that. Just recognize what is responsible for RNA splicing is called a spliceosome, which kind of makes it easy to remember. The confusing part about this is the parts that are cut out are actually called the introns, and the parts that are left in are actually called the exons. And you know what? Someone needs to get sort of chewed out for making the terminology here, because it's basically opposite of what you'd expect. So you would think we would leave in the introns and we would cut out the exons, which would make sense, but it's actually backwards from what makes sense. So exons are actually the parts that are left in, and introns are the part that are cut out. So I apologize on behalf of all scientists for naming it in a way that makes literally no sense. I just always try to remember it's opposite of what you would expect it to be. And I get it mixed up because it is so non-intuitive. So just remember it's opposite, that the introns are cut out and the exons are glued back together. Okay, so I will come back to that actually, because there actually is some significance to that process. I haven't talked about it just yet, but I mentioned that because it'll come back up later after we get through the part on translation. So recognize, put a star next to that if you need to, and recognize I will come back to it. Okay, so following transcription, once we've got our RNA splicing and we've added the poly A tail that I mentioned earlier, the messenger RNA will be allowed to then to exit through the nuclear pores out into the cytoplasm where the ribosomes are located. Ribosomes are going to be a key part of the next stage, which is transcription. So when it comes to transcription, ribosomes will be key players in that process. In order for the RNA to exit, there are specialized transport proteins both inside and around the exit sites, the nuclear pores, where they will allow RNA to bind and sort of slowly coax those RNA molecules out of the nuclear pores. Something that we're not focusing on here, how exactly that works, but they actually sort of have a mechanical process by which the RNA sort of slowly moves out of the nucleus through the pores into the cytoplasm. These figures rarely ever show all the detail, because typically, especially in a eukaryote, you would see the nuclear pores leading directly into the rough endoplasmic reticulum. So ribosomes are located in bulk in the rough ER, which is typically right outside the nuclear pore here, but you also have ribosomes in the cytoplasm for both prokaryotes and eukaryotes. So that part isn't a significant detail to get bogged down on here. The main idea is that the RNA has to leave the nucleus. Transcription occurs inside the nucleus. The next stage, translation, will occur outside the nucleus, and that's sort of the main thing to emphasize here, the locations where each process takes place. Okay, so translation is next. So translation can be defined as the process of converting the sequence of an mRNA molecule into a chain of amino acids. So this is the chain of amino acids. This is the protein itself, but specifically the primary protein sequence. So translation is the first stage of protein synthesis and results in the formation of the primary protein sequence. And when I say it's the first stage, it's really the main primary stage. What happens after the initial primary protein sequence is made is that the protein will later be folded and ultimately shaped into its final form. This can take place in the Golgi apparatus for the most part. So the Golgi apparatus largely is responsible for sort of finishing the protein synthesis. But here we're looking at really the primary aspect of protein synthesis, which is making the chain itself. And this shows, once again, where the ribosomes are located. So emphasized again here, translation is 100% dependent upon ribosomes. So it can only take place where they're located. So this is primarily going to be in the rough ER and throughout the cytoplasm. So you have what are referred to as bound ribosomes in the RER and free ribosomes in the cytoplasm. So if you haven't watched that video, the video I mentioned in the very beginning, the DNA, the transcription and translation animation from McGraw-Hill, you need to seriously stop and go watch that now, because I don't have the ability to show you this process even with illustrations. And so it's very, very important to really understand this, that you go take the time to watch that video. And even if you have watched it, if it's been like a day or two, or you watched it, last week and you're doing this other part last minute here, go back and watch it again and spend just a few minutes, fast forward to the video to the back half of it here, and go ahead and watch that again, because this part here is much more confusing than transcription, and so you need to go watch that video one more time. Okay, but this figure here is showing what that process kind of looks like in terms of the sequence from transcription to translation. So the DNA holds our genes. Each gene is transcribed from the DNA template into the RNA molecule, the messenger RNA molecule. And as we said last time, each of these codons specifies for how to make an amino acid. So during translation, we're actually going to be making the amino acid chain, which becomes the protein. So this chain of amino acids then will look sort of like this as it grows. And after the sequence is produced, it will fold into a three-dimensional shape, and that shape will allow it to take on its function. So here we're looking at how do we end up with this chain like we see here. These are the events that determine, these are the events of translation that determine the protein chain itself. Okay, so the major participants in translation start with the product of transcription, messenger RNA. Messenger RNA actually carries the information for protein synthesis in the form of codons. So the codons are going to be essentially the manual recipe, or however you want to look at it, the information, the manual or the recipe, the information for how to make the protein. transfer RNAs are like little builder molecules, and this is where it helps to see those in action from the video. The video does a much better job of showing the activity of transfer RNAs. But transfer RNAs actually bring amino acids to the ribosomes where protein synthesis occurs. So they transfer amino acids. So they're amino acid transferring RNA molecules. These to me are probably the more confusing participants in the whole process. So I will emphasize those a little bit more here. But if you look back, what is a protein? It's a sequence of amino acids. If you were going to build a protein, you would have to build it one amino acid at a time. Transfer RNAs are essentially molecules that deliver amino acids to the ribosomes where protein synthesis takes place. So each amino acid requires a separate tRNA molecule during translation. Thus, many tRNAs are required. The tRNAs, the transfer RNAs, carry those amino acids to the ribosomes, which are the protein producing factories of the cells. So the ribosomes themselves facilitate protein synthesis by the interaction of mRNA, tRNA, and a variety of enzymes that we will not discuss here for the sake of time. So all these things have to be simplified down. So we're going to focus on the major parts here and skip many of the detailed steps when it comes to the variety of enzymes. So that's one of those things we sort of keep vague. But as I mentioned, transfer RNA is key to understanding how the process of translation works. And depending on what video, depending on what source you use, transfer RNAs can always be shown a little bit differently. So in that video, the McGraw-Hill video, they look a little different than they look here. But transfer RNAs are an RNA molecule that folds into this sort of weird-looking molecule like this here. So this is a flattened out version. Sometimes they call it a hairpin or a clover leaf type molecule. So the molecule is actually a long, single-stranded form of RNA, but the single strand folds back on itself and loops around in these weird shapes, and then closes up over here into this molecule called transfer RNA. So the two-dimensional model sort of looks like this, and the more realistic three-dimensional model looks more like this here. But oftentimes it's more useful to look at more of a two-dimensional model here. But two key areas on the transfer RNA are what I want to actually focus on next. So, take into account the molecule and then focus on the fact that there are two areas. One is called the amino acid attachment site. So these are going to carry amino acids, and they're going to do so right up here on the top part of this molecule where it shows this 3' here and 3' up here. This is called the amino acid attachment site. And so this is the area where the amino acid that the transfer RNA carries will be attached. The other area is called the anticodon region, and it's located down here on the bottom of the molecule, on the bottom loop. This is called the anticodon region. So there are two distinct regions, the amino acid attachment site and the anticodon region. So hold that thought, and we'll go on in the next part to explain more. So during protein synthesis at the site of the ribosome, what's going to happen is that transfer RNA and messenger RNA are going to interact during translation. Ultimately, the messenger RNA contains codons. The codons spell out how to make proteins, how to make proteins by individual amino acids. Transfer RNA contains the region here called the anticodon region. Anticodon loop, as I described it here. One of the most amazing things about transfer RNA is that the anticodon loop will correlate with the codon of the messenger RNA. So the RNA message will correlate right here with transfer RNA at the anticodon region. And what's going to happen is the anticodon region of a transfer RNA is going to base pair with the codon region of the messenger RNA. So messenger RNA and transfer RNA are going to actually interact with each other based upon the specificity between the base pairs of the codon and the anticodon region. Okay, so this is where that video is going to show it to you better. I'm only talking about it here. To visualize it, the video does a much better job. But during protein synthesis, these two must interact. And one of the most, let me go on to the next part here. In order to really understand this process, you have to understand that there's a connection between the mRNA and the codons, and the transfer RNA, the anticodons, and the amino acids here. So recall that codons code for amino acids. One of the most amazing things about it is that the anticodons have to correlate with the codons of the mRNA and the amino acid being carried by the respective transfer RNA molecule. So many things have to happen here. It's astonishing that this is the way life actually works. But the correlation between mRNA and codons and transfer RNA, anticodons, and amino acids, the correlation between all these things is what allows the mRNA to carry the code for protein and the transfer RNA to execute this code during protein synthesis. So many things have to happen here that it gets really confusing. And so I'm trying to break this down as simple as I possibly can. So on the next slide here, we can see here's a transfer RNA. The transfer RNA has the anticodon region down here, and it's going to have the amino acid attachment site up here. This down here is messenger RNA. I should have labeled that. This down here is messenger RNA. And so what we'll see is that during protein synthesis, messenger RNA will come into the ribosome. And we can see a better figure here showing messenger RNA coming into the ribosome. And as messenger RNA comes in, the ribosome is going to position the RNA so that transfer RNA can come in on top of the messenger RNA. And in order for the protein to begin being synthesized, the transfer RNA anticodon region has to match up with the codon region of the messenger RNA. And why that's important is because as we go back to our manual here for how to make protein, the anticodon region will correlate with the codon and carry the amino acid for that codon. So for example, in our manual of how to make protein, UUU specifies the amino acid phenylalanine. Well, in order for phenylalanine to be incorporated into the protein based upon what the messenger RNA is saying, the anticodon has to be complementary to this sequence here. So wherever I have UUU, I need a transfer RNA that says AAA. And if that transfer RNA says AAA, these two will match up and link together during protein synthesis. From there then, that sequence has to then be specific to the amino acid phenylalanine. If I have a different amino acid there, then this message doesn't mean anything. If I have UUU and AAA, but this carries the amino acid for serine or the amino acid for glutamine or histidine, then it doesn't mean anything. I'm going to get something different each time. So specifically, we need a correlation between the amino acid, which is ultimately what we're trying to do, build this protein. I need a correlation here. I need a correlation in the anticodon region of the transfer RNA, and I need a correlation in the messenger RNA. All three of those, at a minimum, have to correlate. And so when they come into the ribosome, this allows a specific codon to interact with a specific transfer RNA, which carries a specific amino acid. All of those things allow the code to be expressed as a protein. Go back and watch that video again. And I'm going to say it one more time. Go back and watch that video again. Watch this part again. Go back here. Watch that video again. And let that sink in a few more times. I promise you, this is not something that's going to make sense the first time you hear it. And so I don't expect you guys to be experts on it in a class like this. We're not biology majors. We're not in biochemistry. We're not trying to study this all semester long. So I'm not trying to get you to speak on it in highly precise ways. But I want you to understand that those are the things that have to correlate here. So for that process to make sense, you have to sort of see that for what it is. And this is one of the most interesting things about life, is that all of this has to take place for even the simplest cells to exist. And so all of these processes correlate with some of the basic core tenets of life, which begin at a minimum with protein synthesis. Okay, so back that up if you need to, and I'd recommend going over that a few more times. The steps of translation, we can sort of break down what's happening in 3 basic steps, and they're going to look a lot like transcription. But initiation. Initiation begins when the messenger RNA binds to the small subunit of a ribosome. The start codon will always occur first in the series. From there, then, the large subunit will bind on top of the small unit and sort of sandwich the messenger RNA in between the 2 subunits of the ribosome. Following that, elongation occurs, and this process begins as the transfer RNAs bring in additional amino acids and the protein chain forms. Termination occurs when one of the 3 stop codons is reached at the end of the protein sequence. There's also a protein called a release factor, which will help separate the protein from the ribosome. Okay, so I'm intentionally keeping this part short because this is an area we could spend a ton of time on. And so watching the video, taking what I just showed you between the transfer RNA and the messenger RNA, then we can sort of abbreviate this part into 3 basic steps: initiation, elongation, and termination, and sort of hopefully get an idea of what's happening there. Big picture. I want a big picture idea, not trying to get bogged down with every single little detail. But initiation requires a start codon, Termination requires a stop codon. Go back to that chart, the manual for proteins, and you'll see specific start and stop codons. Okay, so the primary product of translation, or the product of translation, is a primary protein sequence. The structure of protein in its primary form, which is the sequence of amino acids, the number, the type, and the order of amino acids. So this is the protein in its unfolded, non-three-dimensional state. Later, the protein will fold. Later, the protein will take on a 3-dimensional structure. But as of the point of translation, we're just sort of beginning that process. Now, earlier I talked about RNA splicing. RNA splicing is significant because it allows for what's referred to as alternative gene splicing. Alternative gene splicing is really interesting because what it is it's a way to take a single RNA product and splice it into different mature RNA molecules. So if you go back, and let me just skip back so I can sorry, I cut the mic off there. I want to go back and jog your memory just a little bit here. RNA splicing is a process by which we cut out introns and glue together exons. And so we take a premature RNA and we convert it through splicing into a mature RNA. What alternative splicing suggests is that can be done in variable or alternative or differential ways. So in other words, we can cut out different exons and, cut out different introns. See, I did it myself. Cut out different introns and glue together different exons. And so you can see here in this basic diagram, we have 3 exons: exon 1, exon 2, exon 3. So in one RNA product, we glue together all 3 exons, and we get a mature RNA that looks like this here. In an alternative form, we skip exon 2, and we glue only together exon 1 and exon 3. So we have 2 different ways, alternate or differential ways, of creating a mature RNA molecule that will then be translated differently because the amino acid sequences between version 1 and version 2 will be different. And so what that means is that when we get down to the point of protein translation, the outcome is going to be slightly different. So in this case, a single RNA molecule can be translated differently depending on which exons are included in the mature RNA molecule. The result is that a single gene can be expressed as multiple proteins. So oftentimes the difference between one gene, one splicing version of a protein and the next is subtle. So you typically make slightly different proteins as opposed to totally radically different proteins, but it doesn't take a lot in the shape change of a protein to change its function. So what we're doing is we are ultimately changing the shape and the function of these proteins, and that actually starts in this case by splicing together different exons, creating different mature RNA variants. So this process is called alternative gene splicing. Why I think this is significant is because it helps explain the relative size of our genome and the fact that there isn't an exact correlation between size and complexity. So today, scientists believe that the majority of human genes are variably or alternatively spliced and thus result in variable protein forms. The Human Genome Project has shown us that there is somewhere between 19 and 20,000 protein coding genes in the human body. And the number is not exact. It's below 20,000, last estimate I saw, and somewhere above 19,000. And it continues to be changed. So by the time you're watching this, it may actually be a little bit different, but it's around this number. And so this was far less than originally predicted by geneticists studying the human genome. Nobody really knew how big the human genome was going to be when they started out to study the humans, but most experts predicted that it would be hundreds of thousands of genes, and it was common for people to go higher into the millions. So you look at a human being, the most elegant, complex life forms on Earth, as we like to think, and we see ourselves as much more complex, much more sophisticated. Our brain alone has so much happening compared to other animals and other organisms. I think humans sort of hubrisly and arrogantly assumed we have to just be made of, hundreds of thousands, millions of genes. When the Human Genome Project was concluded, we were sort of left with this humble result, which was that it was actually a very small number compared to what people thought. And what they realized then over time then was that number wasn't exactly related to the complexity. And so if you sort of think of how many proteins you make as a factor of how complex you are, you can think then that the more genes, the more complex. But what they now realize is that the exact number of genes does not directly correlate with the exact number of protein products. With only 19,000 genes approximately, we can produce over 100,000 different protein products. And so this has to do with alternative splicing. And so the original hypothesis that humans are more complex and therefore have bigger genomes is directly not true, because we only have about 19,000 protein coding genes. But the basis for that assumption is true because we can actually make more than 19,000 products due to alternative gene splicing. So it's actually an ingenious trick in Mother Nature to allow a smaller amount of genetic information to code for a larger amount of final products. It really is an ingenious twist in the chemistry of life. And so all I'm asking you to recognize here, going back real quick, is that what is it? It's a way to take a single messenger RNA in a premature form and splice it together to get different proteins from a single gene. So that's what I'm trying to get across there. But on a sort of bigger picture, it allows the human genome to code for more stuff with a smaller amount of genetic material. Last thing, we'll wrap up 2.1 here. All cells in our body, as humans or any other species, all cells have the same DNA. But each cell can be characterized by the types of RNA molecules, i.e. transcripts, that they are making. In other words, what genes are being transcribed and ultimately what genes are being translated. This is referred to as the transcriptome. So the transcriptome of a cell is what RNA molecules specifically are being transcribed. And then that sort of implies what proteins are being synthesized. Okay? So each cell in a multicellular organism carries the same DNA, but what makes each cell differently, ultimately, is its transcriptome, the exact proteins being expressed within that cell. And this will vary dramatically from one cell type to another. So although all cells carry the same DNA, they do not utilize that DNA differently. The transcriptome is sort of a representation of what parts of the DNA they're actually using. So, for example, insulin-producing cells in the pancreas contain transcripts for insulin. So one of the characterizing features of your pancreatic cells is that they produce insulin. And I'm not taking you back into an anatomy lecture here, a physiology lecture here, just sort of speaking very broadly. As you may know, I'm sure many of you know, it's a little bit more specific than that. Some of the isolate cells will help produce insulin. Specifically some of the beta cells. But in general, the pancreatic cells are making insulin. And so this insulin production is not happening, in bone cells. Bone cells and pancreatic cells have the same DNA, but are completely different cell types. And why are they different cell types? Because they are transcribing different proteins. And the proteins are what will ultimately determine the function of that cell and the structure of that cell. Proteins and the products of the genes themselves will ultimately characterize each cell type as being very different. So even though bone cells carry the gene for insulin, they're not transcribing it. It's essentially turned off. And this is one of the things, going back to transcription factors I mentioned earlier, one of the most complex parts of studying genetics is studying the relationship between transcription factors and gene activation. And there's more to it than that. But the transcription factors have to allow the gene to be turned on in order for it to be transcribed in the first place. And so what you find in these very complex ways, not fully understood, by the way, something that scientists are still working out exactly, but what you find is that the transcription of insulin in bone cells never takes place. A bone cell will not produce insulin, period, even though it has the recipe in its cells for how to make it. It doesn't happen. And the same is true for pancreatic cells. Many of the proteins in the bone cells will never be expressed by pancreatic cells. So they carry the same information, but only express specific genes in relation to the characteristics of each transcriptome. So this is ultimately true for all cells of the body. Each cell type in the body, including cancer cells, by the way, will only express certain types of genes. And the specific set of genes that are being transcribed are ultimately what will make up the characteristics. One of the things about cancer cells is that cancer cells often transcribe genes that are not present in the combinations that are seen in other cells. One of the things that's actually been developed in cancer therapy are new ways of targeting cancer cells. And you can target a cancer cell based upon the fact that cancer cells express certain combinations of genes that other cells do not express. Very interesting. So the fact that cancer cells have a unique transcriptome is actually a way to target those if you can identify what that transcriptome looks like and the product of those transcripts themselves. So we're going to finish up in 2.1 with that concept. Very interesting. And in 2.2, we'll go on and take a look at mutation. Mutations, as we'll see here, are changes in the DNA which affect everything that we just talked about. Everything that we just talked about is subject to change by mutation. And so we'll take a look at that in 2.2.